Thumbs up. Was it more difficult this time? Too difficult. Let's see, what are the main functions of the connective tissue? And there are only three types of connective tissues. You've seen these, review your handouts, okay? Everything is from there. So connective tissue, they connect the body parts. Yes, other name. Main functions to protect, to support, and to bind together other body tissues. This is the main functions. I asked you only for three types: bone, cartilage, dense connective tissue or loose connective tissue, adipose, reticular, and blood. Blood we're going to study today. Okay. The next one was explain at least three functions of the skeletal system. So there are many: support the body, protection of the soft organs, movement because they're attached to the skeletal muscles, storage of fat and minerals, remember, and blood cell formation. Today we're going to see the blood cell formation. Okay, you only have to explain three of these. You have five here. Check again. These all from past handouts. Name four main classifications of bones and give one example. This one should be easy. Long, short, flat, irregular. This one was easy? Example, you have many. Long, short, flats, skull, ribs, and irregulars, most likely vertebra, column, the vertebra. Pelvis, they have irregular form. Okay. What is structural and functional classification of nervous system? We recently did this one. Remember? Central and peripheral, right? That is by structure. Functional, sensory or afferent, everything that is coming to your brain, and motor or efferent, everything that is going outside. Okay? So basically this one. And that was it. How was it? Mama?
I need more study at home.
Okay, try to review. And for the final exam, I would suggest you also to review these same questions, okay? Review them again, write down your answer so that you can memorize it. Is Kaito Ozaki here? Or Saito? Saki? No, these are the two who are absent. Okay. Is anyone friends with them? No? Okay. So today we're going to study the blood. This is another tissue, connective tissue. This is a special type of tissue. This is the only one, the only fluid tissue that you have in your body. It's a type of connective tissue, so this would be easier one of your examples in the first question of the quiz. And basically it consists of the blood cells, which we call foreign elements, and the plasma, which is the non-living, the fluid matrix. Okay? So blood consisted of blood cells and plasma. Plasma is mostly contains water. 90% of its content is water. Some characteristics of the blood, you may very well say some of them. Maybe it's sticky, has some metallic taste. Have you ever maybe had a cut and you tasted the blood? Has some metallic taste, opaque, dark color. It has a scarlet red color when it's rich in oxygen. Okay. Usually when you see the figures with the circulatory system, you see some system is in red and the other one in blue, right? So the red one because it's representing oxygen rich, and the blue one because it's representing oxygen deprived. But actually when you see the blood, when it's really red, scarlet red, it's because it's oxygen rich, okay? And when it's dull, it's oxygen poor. It's actually 5 times thicker than water. And it has a very limited range of pH. Okay. The temperature 38 degrees, so it's slightly higher than your body temperature, and it accounts for 8% of your body weight. Volume in healthy men around 5 and 6 liters. Have you most likely you have had some blood tests before, right? So they take blood from you. And what do? We don't see much after they put it in these tubes and they take it away. But usually they would throw the blood and they would put it in a tube. And when they centrifugate, yes, it will separate, right? It will separate the elements of the blood. So you can see, have you seen maybe, because you will work in laboratory, have you had experience?
Health check,
in the hospital.
Blood tube were onto the
probably that's to continue mixing it. But when you are going to separate the elements, you will put it in this machine. There is a centrifuge. So what it will do, it will rotate at very high speed.
I didn't see yet.
Yes, probably you will see it later in the video. But for the rest of you that are not medical related, usually this is what will happen to your blood sample. They will put it in a centrifuge. So they centrifuge this. And the elements will separate because they have different densities. What you see here, the liquid part is the plasma. Okay, so this is the liquid part, mostly water. Then you have like a very thin layer here. This is your white blood cells and the platelets. And the red one is actually the red blood cells, which properly speaking is called erythrocytes. Okay? And this is what we call the form elements. Okay, the cells that are contained in the blood. So we have mentioned the plasma. Plasma makes up to 55% of the whole blood, so a little more than half it's plasma. 90% made of water, plasma. And it's not just a liquid. It contains some substances that are dissolved in the plasma. Some nutrients like glucose, fatty acids, amino acids, vitamins, electrolytes, gases, oxygen, carbon dioxide, some hormones, other proteins, and some waste products from the cell's metabolism. Okay. But the plasma helps to distribute the body heat evenly throughout your body. Okay. You have all your blood vessels and capillaries throughout your body, and your blood is circulating so that you have a very excellent system how to keep your temperature. The plasma has some proteins. These are most abundant solutes in the plasma. They are made by the liver, except antibodies and some hormones. They are not used as nutrients by the cells. Most of the proteins that are dissolved in the plasma are either albumin or proteins related to coagulation, clotting proteins, and some antibodies. So what about the form elements we mentioned? Remember, what are the form elements? Are the red blood cells, erythrocytes, cyte is cell, erythrocyte, leukocytes, these are the white blood cells, okay, and the platelets. Platelets are actually fragments of cells. And from these types of cells, a little less than half of them are red blood cells. Okay, so like 45% of the cells are red blood cells, and less than one is made of the leukocytes and platelets. Here I put some, if you're interested why it's called like that, retro, it means red. It gives you a little idea. Retro means white. So when you see R, B, C, these are the red blood cells, also called erythrocytes. What is the main function of the red blood cells? Why do we have red blood cells? They will carry oxygen, right? We need oxygen. All our cells need oxygen in order to function. So the red blood cells will carry oxygen to all the cells in your body. So they will take the oxygen from your lungs, will carry it to other cells. They will release the oxygen and will bring back carbon dioxide and release it again through the lungs. The red blood cells are small, they're flexible, and they have this shape, like this. It's called like concave. So they are like some discs, and in the center they're flattened. And this increases the area. Okay? An important thing about red blood cells is that they are annucleated. What do you think this means? Sorry again?
No nucleus.
Yes, exactly. They have no nucleus. Okay? And they contain very few organelles. Remember when we study organelles? How do they transport the oxygen? They contain these proteins called hemoglobin. Okay? And this protein will carry the oxygen. And we mentioned that most of the cells in the blood are red blood cells. Approximately like 1,000 red blood cells per one white cell. Okay, so most of them are red blood cells. So we mentioned that oxygen will be carried by this protein, hemoglobin. Have you heard about hemoglobin?
Yeah.
You most certainly yes. Yes? Okay, so what is hemoglobin? Your red blood cell will contain lots of molecules of hemoglobin. But each of these molecules of hemoglobin is composed of 4 chains of proteins. It's called globin chains. And it's not only a long chain of proteins, but they have a specific shape, a 3D shape, very specific. Okay? And you have 4 chains, and each of these chains have what they call a hem group. This hem group. And this hem group will be attached to iron. Okay? And here is where they will transport oxygen and carbon dioxide. But the important thing here is that they will carry the oxygen, but this attachment is reversible. Okay? Otherwise you will not be able to release the oxygen. Okay? So that's very important. It binds reversibly to oxygen and to carbon dioxide. And if I have 4 chains of proteins for one hemoglobin, each of these can carry an oxygen molecule. So one hemoglobin will carry 4 oxygens. Okay? Just so you know, when the hemoglobin is fully saturated, we say this when they have fully bound with oxygen, it's called oxyhemoglobin. Okay, oxy because they are fully carrying oxygen, and it's red color. And when it's not fully saturated, when it's completely saturated with oxygen, it has this purple color, and it's called the oxyhemoglobin. Just for your reference, this is a normal blood amount in men and women. Normal blood amounts of hemoglobin in men and women. And we have this as a reference so that we know when we have lower levels how would we call this condition? Anemia. Okay. If your blood has a decreased ability to carry oxygen, then you are having anemia. There are different levels of anemia that's not relevant to the course. But if we mean that we can carry less oxygen, we may be having less numbers of red blood cells. Okay, so you have less red blood cells. I can carry less oxygen. Or maybe I do have normal amount of red blood cells, but it's my hemoglobin, the problem. Either it's abnormal or deficient hemoglobin. So I cannot carry enough amount of oxygen. Okay. So this is just for you to have a slight idea that there are different types of anemia. Okay. This is just a graph. Types of anemia. If it's a decrease in the number of red blood cells, maybe you cut yourself, you have a hemorrhage, that will very quickly decrease the number of your red blood cells. If your red blood cells, for some reasons, are lysis, this means like they're exploding within your capillaries, maybe because you have an infection. You will also have a decrease in the number of red blood cells. And there are different reasons why you may have decreased number of red blood cells. Another situation we mentioned, if we have inadequate content of hemoglobin in the red blood cells, by different reasons, lack of iron, prolonged bleeding, for example. When women menstruate and they heavily menstruate, they may suffer or be at risk of having anemia, okay? Because they are losing too much iron, more than they are intaking in their diet. Or if you have an abnormal hemoglobin by some genetic reasons. So there are different types of anemia. The other type of blood cell is white blood cells, okay? Usually called leukocytes, leukocyte cells, white blood cells. These represent less than 1% of the total volume of blood, but they are very important in your immune system for the body defense. Okay, the normal ranges are within these numbers, and they're the only complete cells in blood. We just mentioned the red blood cells; they don't have nucleus; they're unnucleated. Okay, and we will see short the platelets; they are not really cells. So they are the only complete cells in the blood. They're able to move into and out of the blood vessels, something that is called diakinesis. They will move, they will go inside as blood cells or outside, okay, when they are carrying their functions. And they respond to chemicals released by damaged tissue, meaning that if you have a tissue that is damaged, it will send some signals, chemical signals, and the white blood cell will come and do their actions. Again, if we have too much or too many white blood cells, it's a condition called either leukocytosis, if you have too much, or leukopenia, if you have too few. Okay, penia is always less than the normal. Okay. Why could this happen? You may have leukocytosis, a lot of white blood cells, because we mentioned they are taking a role in the body defense. So if you have an infection, then you will need that white blood cells to fight the infection. Okay, so your body will start producing more white cells. So the number of white blood cells will increase.
So, is it correspond to the leukemia or leukocytosis?
It's another condition. Yes, leukemia. You will produce, but the cells are not mature and they are not well functioning. Leukopenia, on the other hand, when you are taking especially drugs to fight cancer, they will destroy many of your white blood cells. That is why many people who are undergoing cancer treatment, they are at very high risk of getting infections. The drugs, anti-cancer agents, or some corticosteroids, if you're taking for very prolonged time for some other conditions, they will result in leukopenia. How many types? We usually refer to them as white blood cells, but actually there are different types of white blood cells. The main distinction of them is grouping them in these two groups, granulocytes or agranulocytes, depending on whether they contain visible granules in the cells. These granules are visible when you do the staining, okay? These are the granulocytes. If you see here in the cytoplasm, you can see these like dots. These are called the granules, okay? And we have these three types. We'll see a little more of them later. Neutrophils, eosinophils, and basophils. And you have this other group, granulocytes. If you see here, the little cytoplasm that you can see, it's very clear they don't have the granule, okay? So basically, if they have the granules, they're granulocytes. If they don't, are granulocytes, okay? Who are the granulocytes, the lymphocytes, and the monocytes? So we mentioned the granulocytes, these ones over here. If they have granules in their cytoplasm, it can be stained. Usually they have this nuclei that is not like the previous cells that we have seen, that is a very circular nucleus. In this case, it has like lobes. They're like these circular parts attached with longer areas. So they are lobed. And they include these three types: neutrophils, eosinophils, and basophils. There are some physical differences between these three groups. Mainly, it's the function that they will conduct. Neutrophils are like phagocytes. When you have an infection, an acute infection, eosinophils will react when you have allergies, they increase, or if you have parasites also, they may increase. And basophils, they initiate the inflammation because they contain histamine, which is a substance involved in the inflammatory response. The other type of leukocytes we mentioned is the agranulocytes. Okay, lymphocytes and monocytes. You cannot see here granules. The nucleus is more spherical or like kidney, different from the other ones that are blocked. And you have these two types, lymphocytes and monocytes. So, very quickly, this is how all the blood cells will develop from a stem cell. Okay, the stem cell is like a mother cell that will give rise to all different types of blood cells. In this case, it's called hemocystoblasts. Okay, so they will differentiate and end up producing different lines of blood cells. The lymphocytes, they play a role in immune response, and they're like lymphatic tissue. The monocytes, they're the largest of the white blood cells. You see here, this is the monocytes, this is the lymphocytes. And they function as macrophage in tissues, and monocytes are important for chronic infections. Okay, so they will increase when you have a chronic infection. When you have a blood test taken and the doctor see your results, sometimes by the increase of some of these lines, they may suspect of different conditions. Either if you have parasites and you are maybe not experiencing too many symptoms, or if you have an infection. So this gives clue to part of the diagnosis, will help for the diagnosis. And you have heard about platelets? Yes? These are actually not cells in the strict sense, okay? They come from these cells called megakaryoblast, but they are fragments of cells. They are fragments of the megakaryocytes. And why are they important? Because we need them for the clotting process. What is the clotting process? To create a clot. When do we need a clot?
Wound injury?
Yes, if you have an injury, then you will need a clot. Otherwise, you will bleed until you die. Okay? So it's very necessary. Hematopoiesis is the process of formation of blood cells. Okay? And where does this take place? In the red bone marrow. Okay? Only in the red bone marrow. Usually in adults, red bone marrow is in the flat bones. Where do we have flat bones? Skull. Okay. In the pelvis, in the ribs, in the sternum, and proximal epiphysis. Humerus. Where is the humerus? The arm. Femur is in the thigh. Okay. When you are born, actually you have red bone marrow in your whole skeleton. But while you are growing up, it will be replaced with osseous tissue, and you will only end up with specific places or limited places with red bone marrow. All blood cells are derived, they are all coming from a common stem cell. Okay, from a common ancestor, like a mother. And it's called hemocytoblast. Okay, this is a very simplified way of representing the lines of the different blood cells. There are some intermediate stages, but it's not very, maybe it's the next, oh yeah. There are different stages in between these with some other names, but it's not really important for you to memorize all of that. Just for you to have an idea, they all come from a stem cell. The stem cell will give rise to different lines of these different blood cells. Okay, the red blood cell, the granulocytes, this one the monocytes, granulocytes, platelets, and the other granulocyte and lymphocytes. What about the formation of the red blood cells? It says unable to divide, grow, or synthesize proteins. Why would that be?
Because they have no nucleus.
Yes, that's right. They have no nucleus. So the lifespan, the time that they will be alive, it's around 100 and 120 days. Okay? And they will be disposed of, they will be eliminated in the spleen or in the liver. This hem group that we mentioned that is present in hemoglobin, it will be degraded to bilirubin. And the bilirubin will be secreted in the intestines. in the form of stercobilin. Stercobilin is actually what gives the brown color to your stools. Okay. The lost cells, if we have lost cells of red blood cells, will be replaced by division of the stem cells. So if I have a low count of red blood cells, my stem cell will have to become more active. This is detail. If you see from this one, this gives rise to erythrocyte, which is the mature form of the red blood cells. Before this one is reticulocyte. Okay, just in case if you're actually reading the handout, reticulocyte is a previous stage or level of the erythrocyte, and if you see here, it still has some of the organelles. So the young red blood cells are called reticulocytes because they still contain rough endoplasmic reticulum. They will go from hemocytoblast, from this phase, to be a mature red blood cells in approximately 3 to 5 days. Okay. So it will take time. That would mean also if you're having a low count of red blood cells, really severe, you cannot really wait for your body to raise the level. Then you will need to complete the level of red blood cells, maybe by transfusion. Okay, transfusion, you can transfuse either only red blood cells or plasma or platelets or the elements that you are needing. How is the formation of red blood cells controlled? By the production of a hormone. The red blood cell production is controlled by this hormone called erythropoietin. Okay? And this is produced in the kidneys and in the liver. Okay? Erythropoietin is a hormone producing red blood cells. Where is blood cell produced? In the bone marrow. So the target of this hormone must be the bone marrow. Okay? And here you have a nice example of a negative feedback. Remember when we were studying homeostasis and negative feedback and positive feedback. If you have a low level of oxygen in your blood, your kidneys will detect this and they will release erythropoietin. Erythropoietin will act on your bone marrow, increasing the levels of your red blood cells because the red blood cells will carry the oxygen. And therefore increasing the level of oxygen. Right? The way it's controlled, the production of the white blood cells is different from the red blood cells. The white blood cells are stimulated by some factors. It's a group of factors, but altogether they're called colony stimulating factors and others called interleukin. They will stimulate also the red marrow. They will enhance the ability of mature leukocytes. And they are released in response to some inflammatory chemicals or some bacteria or some toxins so that your body can be able to defend itself. What happens then when we are losing blood? We have to stop the bleeding, right? So hemostasis means prevention of blood loss. Yeah, there are different steps through which your body will control this loss of blood. Imagine that you cut one of your blood vessels, okay? If you don't repair it soon or quickly, then you will continue losing blood. The first will be the constriction, the vascular constriction, okay? Then, this is very rough, the formation of a plaque, a platelet plaque. So the platelets will be the first ones to come and to create this plaque. The formation of a clot, your blood starts to coagulate, and then the growth of fibrous tissue into the blood clots to close a hole in the vessel. This is a figure with the explanation. Step 1, if you imagine you have a rupture here, step 1, then you have a vascular spasm, the smooth muscle will contract and will end up in a vasoconstriction. But this is not enough to stop the bleeding, especially if it's big, right? Step 2, there will be a formation of platelet plaque. The injury to the lining of the vessels expose some fibers, collagen fibers, that will make the platelets to adhere to this area. The platelets will release some chemicals and will call for more platelets and form a plaque. And step 3, there will be a formation of fibrins that will form a mesh. Okay, it's like a mesh that will trap the red blood cells and platelets and will form the clot. There are all the steps for the coagulation is actually very complicated, and there are many factors involved in these. We will see only a very rough idea of how this works. Okay. Normally the blood contains some factors that are called procoagulant factors. They will stimulate the coagulation. And we have anticoagulant factors that will inhibit the coagulation. So when the vessels are damaged, the procoagulants will increase their activity and they will form the clot. And the formation of the blood clot will involve a very complex series of events, of chemical reactions, and they include different clotting factors that they have to be present in plasma. Okay? This is the list of the factors. You don't need to memorize all the list. Okay? But this is just so you know that there are different factors involved in coagulation. A very summarized idea of what is happening. For example, you have a trauma, you have an injury to the vascular wall. These will stimulate the intrinsic path, which is called the intrinsic path. Or you may have some trauma to the blood, or the blood will come in contact with some cells, endothelial cells, or some collagens that are outside the vessel. And this is called the intrinsic path. No matter which of these paths the injury comes, it will start a cascade. A cascade is something that happens, this will trigger another reaction, this reaction will trigger another reaction. You will start a complex cascade of chemical reactions where all these coagulation factors are involved. So this prothrombin is continuously formed in the liver. And in the presence of vitamin K and calcium, the prothrombin activator will actually make the inactive state of prothrombin to change into an active state called thrombin. This thrombin, in the presence of calcium, will make the fibrinogen to start producing the fibers necessary to produce the clots. The liver produces most of the coagulation factors, and many of them will require vitamin K to produce all these reactions. So you know that if you have liver failure or a problem with your liver, or maybe you have a deficiency in vitamin K, or if you have abnormal low levels of calcium and so on, then all these cascade of reactions will not take place. And then in the end, coagulation will not take place. And this could put at risk even your life. Summary of what is happening with coagulation, what I already explained. The platelets and the damaged tissues will release the chemicals. This will initiate, will start the reactions that will end up in the formation of prothrombin activator in the presence of calcium, and this prothrombin activator, prothrombin in the plasma, will convert to the active form of thrombin. Thrombin in the presence of calcium will convert the fibrinogen into an active state, an insoluble fibrin. And these will create these threads, the mesh, that will adhere to the damaged tissue and will trap the blood cells and platelets. Let's see very quickly some disorders of hemostasis. Disorders related to the clotting, for example, thrombus. Thrombus is a clot that will form in intact blood vessels. So up to here we have been seeing how the blood clot forms because there is a trauma to the blood vessels. But what happens when this is in intact blood vessels? A clot forms, and this is called thrombus. Okay? If the thrombus is large enough, it can actually prevent the flow of the blood. This makes sense, right? If you have the blood vessel and you have a clot, a thrombus creating and preventing the blood flow, then you will have some organs not receiving oxygen and nutrients and so on. Some example will be if you have a coronary thrombosis, a thrombus in the coronary arteries, then you will have part of your heart muscle not receiving blood. These will lead to maybe a heart attack that could be fatal or not. If the thrombus breaks free and travels through the circulation, we call it embolus. Okay? So this is a thrombus that breaks away and flows freely in the bloodstream. And this is very dangerous because they can later clog some vessels along the way. Some in very critical areas, like your brain. Okay? An example of this is when you have a stroke. Maybe you had a thrombus created somewhere, and when it's circulating in your body, it got stuck, it reaches your brain. So that part of your brain is not receiving blood flow. It's not receiving oxygen, nutrients, and therefore the tissue in that area will likely die. You may end up having a stroke, disabilities, or even die. So those were disorders of clotting. Okay, clotting forming here and there, or maybe thrombus breaking free. But you can also have some problems that make you bleed. Okay, one goes to clotting too much, the other one goes to the other extreme, bleeding disorders. Bleeding disorders that could be inherited, you may be born with them, or you can acquire them. We mentioned because you have deficiency of vitamin K, maybe you have problems with your liver, you cannot produce coagulation factors and so on. Now let's see. Do you all know your blood group? Do you? There are actually over 30 common red blood cells antigens in the humans. But the antigens that we most know is the ABO. Either you're A, you're B, you're AB, or you're O. What type of blood? And RH blood groups, either if you are Rh positive or if you are Rh negative, right? I guess all of you maybe are positive, Rh positive. You don't know.
I don't know.
You don't know. Yes. If you are Rh negative, most certainly you will know, because they are not so common, and you will see later on it's because they're not common, you will not have easily matches if you need blood transfusion, and Rh positive cannot, in principle, give you blood. So let's first see what is an antigen and what is an antibody. We will from here on mention a lot about antigens and antibody. Okay? These two systems, ABO and Rh, are the ones that causes the most vigorous, the most strong reactions when we have transfusions. That's why we usually speak in terms of ABO and Rh when we talk about our blood type. So what is an antigen? Any idea? No? What is an antibody then? We produce antibodies, right? And we produce specific antibodies against an antigen. Okay? And an antigen is a substance that our body recognizes as something foreign. This is not part of my body. That is what is an antigen. Okay? But we will see here with the blood groups and the blood types, when we say foreign, it means that my body does not recognize this as mine, but maybe your body recognizes as his. Okay? Okay, in this case, the blood groups, the membranes of the red blood cells have some antigens, okay, these substances that the body will recognize as foreign, and these are genetically determined, okay, so you're born with these antigens in the surface of your red blood cells, and these will identify the person as unique, okay, you have a unique set of these antigens, and each of us tolerates our own cellular antigen. So we have these antigens in the surface of our red blood cells, but my body recognizes this as my own. Okay? The antibodies present in the plasma will attach to the red blood cells that have the antigens different from those on the recipient's red blood cells. The very basic idea is who could donate blood to you. Think a little in those terms, okay? If I'm A, who can give me blood? If I'm a B, who can give me blood? Who can I give blood to? If I'm O, and so on, okay? That would be an exercise for you at home. So the binding of the antibodies will cause the foreign red blood cells to clump, to agglutinate, and they will clog the small vessels, and the red blood cells, the foreign red blood cells, will be ruptured, we call it glands, and the hemoglobin will be released to the bloodstream. So let's see first in the ABO group. The ABO is basically if they have or if they don't have antigens type A and type B. Okay? They are based on the presence or the absence of these two antigens. The absence of both antigens will result in a type O. So someone who has type O does not have any of these antigens. Correct? The antibodies formed during infancy against the ABO, the antigens not present on your own are red blood cells. So if I am O. Yes, thank you. Okay, let's see an example. A baby with blood type O doesn't have antigen A or B. Okay, so this baby will form antibodies against antigen A and B. Okay, this is to protect himself. Yes? You can have this, this is a chart. You can see it more carefully at home. It's here type A, type E, type AB, and then type 0. Remember type O. Type O doesn't have any antigens in the surface. Okay? Type AB will have antigens A and B. B will have antigen B, and A will have A. There is a test very quickly by which you can find out what kind of blood type you are. Just putting a drop of your blood with some serum containing antibodies. Okay, the other system by which we describe the blood groups is the RH. RH positive means that the red blood cell will carry an RH antigen. Again, if I have the antigen, I'm positive. Where were we? RH blood groups. Okay, so RH means that the red blood cell will carry the antigen, RH antigen. Anti-RH antibodies are not automatically, you're not born with these. They're not automatically formed, they're not present in the blood of those who are RH negative. And this is why I mentioned, in principle, if you are RH negative, you shouldn't receive blood transfusion from positive donors. Problems can occur if you mix RH positive in someone who has RH negative. Okay? But actually the reaction will come in the second transfusion, the third transfusion, and so on. If a RH negative person receives a blood from someone who is positive, shortly after the transfusion, this person's systems will become sensitized, will start producing the antibodies. And the second time that this RH negative person receives a blood from RH positive person, from that moment on will start hemolysis or breaking down the red blood cells which are foreign to this body. Okay? When is this especially critical? Imagine if you are Rh positive and your wife is negative. What will happen if you have a baby who is positive? Okay? Imagine that the father was Rh positive, but the mother was negative. And they had a baby, and the baby is Rh positive. Do you think it will be risky for the baby? They will have some problems? So the first time that this woman becomes pregnant of an Rh positive baby, maybe there is no big problem, right? Because we say at that moment she's not sensitized yet. But the Rh negative mother carrying the first baby who is Rh positive, the Rh antigens from the fetus can enter the bloodstream of the mother during delivery. Okay? So if she's negative and the antigens are coming through her bloodstream, she will respond, her body will respond to these fetal Rh antigens, and the mother will start producing the antibodies against the Rh. Okay? So far so good, everything's okay. But if this woman who now is sensitized and has the antibodies becomes pregnant again with a baby who is again positive, then her antibodies will start identifying as a foreign, the baby Rh as something foreign. So if the woman becomes pregnant with another Rh fetus, her antibodies, anti-Rh antibodies, will cross the placenta and start damaging the blood cells of the baby. Okay? So it is very critical when the mother is negative and has the first baby Rh positive to treat this mother so that she does not sensitize. Okay? That her body does not start producing antibodies. Have you ever donated blood, or have you ever received blood transfusions?
No.
No. No. So there are some reactions to transfusions. They can cause fever, chills, nausea, vomiting. But the most devastating consequence when we have severe reactions is that the hemoglobin molecules are free, and they may end up blocking your kidneys. These will result in kidney failure, so your kidney may not be able to work, and it could cause death ultimately. Okay? If we lose blood, if we cut, all of us, we have had small cuts. Usually we don't feel much sick, right? Unless you are terrified of blood and maybe you pass out from just watching. But usually you may start feeling a little weak. What is pallor? These were here. If you lose between 15-30% of your blood, you start feeling weak and pale. Okay, pale. So your white maybe starts becoming white. But if you lose more, you may end up dying. You will go into shock. As I mentioned earlier, the fastest way to replace blood loss will be transfusion. Okay? But if you transfuse blood, it has to be the same blood group, the same RH. The blood of donors is mixed with anticoagulant, blah, blah, blah. Okay, this is not really that, right? So what I want you to think about, maybe we can do it in the 10 minutes that are left. Think about, none of you knows your blood type, right? So do a grid, okay? If I'm type A, if I'm type B, if I'm type AB, if I'm type O, who can I donate blood to? And from whom can I receive? If you are blood type A, what blood types can you receive? If you are blood type B, what blood types can you receive? The same if you are AB or if you are O. Why don't you take 10 minutes maybe and discuss? No? You want to do it alone? 10 minutes to think? Yes? Make your table. Type A, B, AB, O. How do you call this group? How do you call them? It's the first time you see them?
Yeah.
Oh, yes? Yeah? No, right? How do they call them? How do you call the group? Why are they doing this?
Promoting Yoshida residence Matsuri festival.
Oh, why? I don't understand. Yes, I saw them last year too, and I don't understand. Yeah, because they come in here, they disrupt the class, and then they request not to be photographed. That is very selfish. You know? I complain to the office, and they say there's nothing we can do. How come? I would expel them. Okay, so we have an answer.
Yeah.
Yes? So let's say you're type O. This is the easiest one. Maybe it's easier to understand too. If you're type O, who can you receive from? Who else?
Only O.
Yes, that's right. Only O. Okay? So imagine if this is my red blood cell and you give me one of these, they already have antigens in their surface. So that will make my body to create antibody. Okay? Because they identify this as foreign. So no, I don't need these. I only can work with these. Very good. And how about if I am AB? Who can give me blood if I am AB?
So who can give to AB?
Yes, from whom can I receive?
AB and O.
A, B, and O. So everybody can give me,
Everybody.
right?
Yeah.
And A?
O and A.
Yes. And B?
O and B.
O and B. Yeah, that's right. Think of if you have to memorize, think of O as the generous one. Okay? O is the generous. O gives to everybody, but O only receives from O. And AB is the selfish one, because AB cannot actually give to other than AB. But AB will receive from everybody. Okay? Thank you very much. Don't forget about this one, okay? Which type has which antigen and what kind of antibody they will be producing. Okay? Remember, A has antigen A and produces antibodies against B. Cannot produce A, otherwise he will kill himself. Okay? So, with more time, you will read this at home. If you have any questions? No? Okay, then we'll see next week. I think next week we start with presentations, right? Who is the first one? Heart. You.
Yeah.
Okay. Wait a minute. You too?
No.
No. Okay. Yeah. Only one person. We have one student who is not coming. And he's supposed to do the other part. So if he doesn't come until then, it's only you. Okay? Just do your part. And that will be okay. And I will cover for the physiology of the heart. Okay. So thank you very much. If there's no more questions, quizzes, was it okay? Too difficult? No? Please study from the past classes. Okay? Yes. Thank you very much. I'll see you next week.
Thank you.